Namaskara Karnataka, I am Miss Gopika, your own biology master teacher. So welcome all of you to another amazing session of most important questions from human health and diseases for my dear students who are preparing for PUC board, right? Because PUC board, what happens is you do not have the time at the end, to prepare, and you will be confused where to find questions. So don't go anywhere. Our channel is there. Just subscribe to the channel, like the video and share it with all your classmates, because I know that if one of you share one video, it can help, and also it can help a lot of students. Also, it is like, giving us some love as well, right? So I hope we are live and I am audible and visible to all of you. Let me just refresh. Yes. Yes, we are live. Very nice. Okay, so students, on your request, I had posted molecular basis of inheritance, but the response on that, I'm a little disappointed, because molecular basis of inheritance is a 7 hours chapter. So I have done 2.5 hours of main portions. Again, we have one more session where we'll be doing the rest of it. So I want all of you to go and watch it today itself and finish that chapter, because that chapter has in your KCET 5 to 7 questions. That means it's a very high weighted chapter. And for those students for boards, I do not have to say, because molecular basis of inheritance always fetches a lot of marks. Okay? Yes. Chalo, let's start the class. Hi, Riyaz. Now, we will start with the first question. State the functions of mast cells in an allergic response. So, students, all of us have allergy, right? One or the other way, we all get allergies. It could be because of dust. It could be because of, some food items like walnuts, peanuts. It could be because of pollen grains. Allergies are very common. Now, here the questions ask that state the function of mast cells in allergic reaction. So, remember that an allergen, allergen is going to come. It is going to cause you an allergy, and when an allergy happens, immediately mast cells in our body will be released. Okay, mast cells, students, have a capability of releasing histamine. Okay, it has a capability of releasing histamine. This histamine, what it does is very important for you to know. This histamine is a vasodilator. What is, ma'am, vasodilator? Students, vasodilator is nothing but, normally your blood vessels are like this. Just let's imagine, this is how your blood vessels are normally there. Okay, this is the diameter, this much blood can flow through it. The moment histamine gets released, vasodilation happens, leading to more blood flow. Okay, here let's say, here I think difference is too less, so we will make this a little more cutie. Okay, I'm not talking in the cases of length, I'm talking in the case of dilation. Okay, so let's say, students, here what happens is vasodilation will happen. So here, if only this much blood could flow, here more blood will be able to flow, because of which this histamine release will lead to vasodilation. It also shows that you have an inflammatory response, because the blood is going to flow and you will automatically start seeing redness. Right, you will start seeing redness and rashes all around your body to identify that you have an allergic reaction. Otherwise, how will you know you have an allergic reaction? Okay, an inflammatory response is going to happen. So what exactly mast cells are going to do? Mast cells release histamine, which causes inflammatory reactions in the body. Very easy question, no? 1 mark question, so it's very important for you to remember. So quickly like the video na bega like ಮಾಡಿ ಆಮೇಲೆ start ಮಾಡೋಣ. Okay, start ಮಾಡೋಣ class. Yes, so students, mast cells release histamine, which causes inflammatory reaction in the body, is a 1 mark question, and this question was asked in 2019, so it is important for you to remember. Okay, next question. Name the primary, secondary lymphoid organ. Name the primary and secondary lymphoid organ. Now, lymphoid organ, I told you that primary organ is like a military, like a training camp. Okay, like a training camp. Student, writing is visible, no? Okay, writing is visible, no? 1 second. Yeah, just give me a minute. I don't know what happened. Okay. Yes. So look here students, it's visible, right? Yes. So it's like a training camp, and secondary lymphoid organ is like a battlefield. Is like the battlefield. Why I'm saying this? Because in the primary lymphoid organ, actually we are, doing the production, basically we are doing the training. And the battlefield is where, students, the battlefield is in a different place where they will be encountering the antigens that is going to happen, right? Very good. So primary lymphoid organs are organs where the B cells and the T lymphocytes are formed. Okay, and mature, acquire antigen specific receptors such as bone marrow and thymus. So your primary lymphoid organ is your bone marrow and thymus. And remember this also, since we are in this topic, I'm going to add on to this also, students. Your B cells, okay, B cells production, maturation, everything happens in the bone marrow. Okay, bone marrow. But your T cells, that is not the case. Okay, T cells maturation will happen in the thymus. Okay, maturation will happen in the thymus. Thymus, clear? Maturation will happen in the thymus. This is very important for you to remember. B cells, bone marrow, okay, everything happens in the bone marrow, like the formation, production, as well as the maturation. But D cells maturation will happen in the thymus. This you remember. You can remember easily, B for bone marrow, T for thymus, like that you can remember. Now, but it's not the technical derivation of it, okay? Yes, such as bone marrow and thymus, this is the primary lymphoid organs. Now, secondary lymphoid organs, we will draw a line for this. Secondary lymphoid organ, location where mature lymphocytes resides, including your lymph nodes, okay, including lymph nodes, spleen, okay, spleen, tonsils, Peyer's patch, and mucosal surfaces. This mucosal surfaces, here they are talking about your malt, okay, mucosal associated lymphoid tissue, where 50% of the lymphoid organ will be with having this malt, okay, mucosal associated lymphoid tissue. Now students, listen here, this secondary lymphoid organ is the place where this, this B cells and the T cells will be actually encountering your antigens. Encounter the antigens. Clear or not? Okay, clear? Yes, please put a thumbs up. Yes, Kush, understood. Very good. Students, all these notes will be available on this Telegram, Biology by Gopika, as well as our official Telegram, V Karnataka, KCT and boards. So do not forget to join the Telegram as well. Okay, chalo. So this question is clear, no? Easy question, students. Many times the questions have been repeated. So what I have done is, I have gone back to previous year questions and I have picked questions that have been repeated and I've changed the pattern, okay, and have put the answer so you people know what you're supposed to write, okay, because if this is a 2-mark question, you do not have to explain a lot, but if it's a 3-mark question, you have to write all of this, that B cells, T cells, production, maturation happens here, B cells, T cells, what is happening, all that you have to write, okay. Chalo, let's move on. Now, what are the various routes by which transmission of human immunodeficiency virus takes place? This question has repeated so many times, okay, repeated so many times, students. That is, the question is this: what are the various methods by which transmission of HIV, AIDS causing, no, AIDS causing virus got transmitted? How is it possible? First one, sexual contact with an infected person. That means you have a sexual intercourse with the infected person, then you will end up having the infection, okay. So sexual contact with an infected person through blood transfusion, because I told you that AIDS is not passed merely by touch or handshake. AIDS will only be passed by a fluid exchange like your blood, semen. This is where the exchange can happen, okay, or where the syringes and needles are shared. Okay, so here through blood transfusion. Second thing is by sharing needles, often among intravenous drug abusers, plus students in tattoo cases. Okay, plus in tattoo cases, because what happens when we do tattoo, what really happens is the needle will be poked over and over, right? That time there are chances that some blood will come in contact and the needle will come in contact with the blood. So if that immediately is transported to another person, it will happen, but do not right? HIV is transported by mosquito. Mosquito bite does not causes. Okay, mosquito is not a vector in carrying HIV. Mosquito will not do the job. Okay, because of the fact that mosquito will digest the blood meal, so it will digest the, and you know, no virus, if it does not have a host, it is like one, it will be like one silent cat. Like, you know, in school and all, we tell, no, my god, this my kid is a silent cat. Your parents used to ask, no, when you go to school, my kid is silent, are you serious, ma'am? Yeah, ma'am will be like, yeah, I've never heard her talk, I've never heard him talk, but in home to violent. So like that only this you know, HIV is like this, or virus for that matter. Until it reaches the host, it is very silent. The moment it reaches the host, it becomes violent. Okay, it becomes violent. So from mother to child through placenta, this also can happen. That is, from, if the mother is HIV positive or AIDS positive, it can come to the child as well. Okay, students, please understand this also. I have explained it multiple times before also. HIV positive does not mean AIDS positive. Okay, HIV positive over a span of, could be 5 years, could be 10 years, could be 15 years, over a span of years, they become AIDS positive. Okay, this is an good information for all of you to remember. HIV positive does not merely means AIDS positive. HIV positive over a span of years only becomes AIDS positive. Done, done? Easy, no? Yes, okay. Next question. How is cancerous cell visible, right? Yes, it's visible, no? How is cancerous cells different from normal cell? Okay, how is cancerous cells different from normal cells? Answer, normal cells remain adhered to one another. So remember like this, students, let's draw one you know, tumbler. One tumbler like a little patra, no? Like that, one tumbler we'll draw. This is one tumbler. Okay, and this is another tumbler. Okay, 1 and 2. So if you look here, you will notice that this one is, let's say this is the normal cells. Okay, this is the normal cells. It will take some time. Yes, these are the normal cells. Let's say this is normal cells. Now what happens in normal cells? Okay, let's say Riaz is there in class, Kush is there in class, Yasmin is there in class. Okay, so these people are like normal cells. So when they come in contact with each other, I'm telling them, you people have to sit in the sofa. That sofa has only space for 3 people. So Riaz will sit, he will give space for Kush, he will also give space for Yasmin. Now when they come in contact with each other, they know that they have to adjust and sit because the sofa place is only that much. So that is how normal cells work. But cancerous cells are not like that. They are like, no place in sofa, it's okay, I will sit on your lap. So like that kind of a people, because they do not, or they lose the ability of contact inhibition, which is a very, very important topic for you people to remember. They lose contact inhibition, students. Contact inhibition means normal cells, when they come in contact with each other, they will understand that they have to stop multiplying. Okay, they have to stop multiplying and they have to be, okay, I have to stop it because I came in contact with the next cell, it's over. But these cancer cells are not like that. They will keep on multiplying. Let's draw an example, students. So this is our normal cells. Okay. Now here we have cancerous cells. Cancerous cells. Like this, they will keep growing over everywhere. They will grow. They do not have a proper way of growing. So these people, so normal cells adhere to one another. They have a definite lifespan. After some time, they have to undergo, they have to die. Okay, no other choice. They have to get degenerated and they have to die. As some old cells die, they are replaced by new cells, which arises by cell division. Okay, cancerous cell, whenever there is a breakdown of any regulatory mechanism, a cell develops the ability to undergo uncontrolled division. So here, first thing is they lack, okay, let me just write that, students. This is very important because this question also I've seen a lot of time. They ask you cancerous cell, they ask you what is normal cell. So here, first point is they lack ability of contact inhibition. Okay, contact inhibition. Okay, contact inhibition. Second thing is they do uncontrolled division. Uncontrolled division. Okay, uncontrolled division. And students, third thing they do is metastasis. Okay, they will keep growing, they'll keep growing, and most thing is metastasis is seen in your malignant tumor. What they do is, from, let's say, from one place, Vinuta and I am going, okay, Vinuta will go to one place and she'll make friends there. But I'm not like that. I will just go to one place, I will just stay there. I will not move from that place. Vinuta is going to one place, let's say location A, she'll make friends there. She'll go to location B, she'll make friends there. Location C, she'll come, she'll make friends there. This is called as metastasis. That means wherever the tumor is going to go, it is going to start growing another tumor there. Again, it will move from there, grow another tumor, come back here, grow another tumor. Understood? Metastasis. Okay? Clear or not? So these are the three main topics of your cancerous cell. Next, what some of the 1 mark questions also, let's see. Okay, what do you mean by health? Health is a state of complete physical, mental and social well-being. So that's what I told you. Just by mere physical fitness, like some people don't know, I have six packs, I have biceps, I have triceps. That is not health. That is just physical fitness. Okay, that is just physical fitness. When you are socially good, that means like how you talk to another person, how you behave with another person. And when I say that you are mentally also doing well, like you're not in depression, you're not crying every day, you're not, complaining about the smaller minor things in life. So that means what, you're mentally also in a very good state, you're socially also is in a good state. That is what is health. Okay, now name 2 non-infectious diseases which are the major cause of mortality, that means for death to happen. Here the answer is hepatitis B and AIDS. So students, the problem with AIDS that all of you should understand is this AIDS, people do not die because of AIDS. People die because of opportunistic diseases. That means, there are some people in life also that the moment we are in a bad phase or moment we are in a good phase, they come into our life. So opportunistic means what? When the opportunity is right, I will attack you. Okay, so same thing, opportunistic diseases. That means they know that a person who's undergoing AIDS, okay, or person who's affected by AIDS have very less T cells. T cells are related with cell-mediated immunity. My students already know this. Okay, cell-mediated immunity. So, when the cell-mediated immunity is affected, T cells are reduced in number. When T cells are reduced, that means your immune system is not working that well. That means your immunity is reduced. So, in AIDS person, when immunity is reduced, automatically opportunistic diseases will be waiting to affect this person. And students, the person dies of these diseases but not because of AIDS. Understood? Clear? This concept is not clear to many of the students. They think, oh ma'am, AIDS only, AIDS causes the disease. No. Opportunistic diseases, that means the ones who are waiting, telling that, okay, wait, T cells have become less now, immune system is going to be, immunity is very less, now let me go attack, then pakka, it is going to spread, it is going to become infectious. Okay, yes. What are pathogens? All of you know, the organisms which have the ability to cause a disease is called as a pathogen, right? So students, disease means what? Disease means an uncomfortable situation, right? I am not at ease. Uncomfortable situation. So a pathogen is an organism that can cause a disease. Clear to all of you? Yes? Done. Can I get a thumbs up in the chat box? If all of you understood, then we will move forward. Yes, tell me. Clear? Click clear. Are you all enjoying biology classes? I'm not able to get a track of if you people are enjoying or not, so that you people should definitely comment and let us know how you're feeling. Okay, what are the things that you want us to change? What are the things that you want us to do? Okay, so this way we will be able to have a smooth journey in this channel, right? Yes. And you people are not sharing, okay? Vinuta, Riyaz, Kush, all of you have to share it with your friends. Okay, share the channel link with your friends and ask them to subscribe and stay in the channel. Okay, we are going to show results this time. Okay, good. So expand PMNL. Expand PMNL, that is students, polymorphonuclear leukocytes. Okay, polymorphonuclear leukocytes. PMNL has come from your body fluids and circulation actually. Okay. So from there, okay, enjoying, very nice. Polymorphonuclear leukocytes. So students, here they can ask you full form of NACO. They can ask you full form of HIV. They can ask you full form of AIDS. So all this be ready, okay? All this be ready. Any of this they can ask you. NACO, HIV, AIDS, any question they ask, you cannot fight with them telling you, why did you ask me and all that, because I didn't study. No, it is there in the textbook though, they can ask, okay? Yes. Question number 11, name the common fungal disease that infects man, okay? Common fungal disease that infects not only like everyone is ringworm allergy, right? Ringworm allergy, and it is caused by the genera of Trichophyton. Okay, Trichophyton, Epidermophyton and Microsporum. So you can just write, okay, Trichophyton, etc. I'm writing as of now. So these people, Trichophyton, Epidermophyton, Microsporum, all of these people causes ringworm allergy. Students, ringworm allergy is mainly happening because of sweat. Okay, like you share towels where the sweat is exchanged, or you go to gym and you use the same carpet that someone else used and just got up, you do not sanitize it. If that person has any kind of allergy, you will also have. And immediately after the gym, if you do not shower, you repeat the clothes, all these cases you will get ringworm allergy. Okay, name the vector responsible for chikungunya. It was asked in 2008, Aedes mosquito. So students, I'll give you a tip to remember this, just not a tip, just remember this, okay? Remember Culex mosquito, one second. Yeah, so different mosquitoes, okay? Mosquitoes also have more, see, celebrity feeling it has, different types of mosquitoes. 3 mosquitoes you remember for this chapter, more than enough, okay? First one is Culex mosquito. Students, this Culex mosquito causes elephantiasis. Elephantiasis. Okay, imagine one kutti mosquito. If elephant sees, no, elephant will be like mosquito. That kutti mosquito can spread elephantiasis, okay, which is a chronic disorder. Then you have AIDS. Sorry, Aedes. AIDS is in my head. Aedes mosquito, which can cause dengue, chikungunya. Okay, chikungunya. Then you have Anopheles. Anopheles mosquito, which can cause you malaria. Okay, malaria. Clear? Just remember these 3 mosquitoes. Okay, very celebrity people they are, so don't ignore them. They are celebrities, okay? You have to remember them and you have to yes. Easy, just make a note of it, okay? Just make a note of it. Yes. So students, actually when we teach dengue in depth, the mosquito's name is actually Aegypti. Okay, so here Aedes, most of them tell Aedes for chikungunya and dengue because we do not go into the depth. So as of now, we will just write chikungunya and I will also write Aegypti. So, some students can at least use it for adding their knowledge. Okay. This is not mentioned in your NCERT. Is dengue actually speaking, but everyone tells Aedes and Anopheles only all the time. So these 4 are the mosquitoes, okay? But then Culex, Aedes and Anopheles you cannot forget. Culex is elephantiasis, Aedes is chikungunya and Anopheles is your malaria, which is very, very important because malaria is digenetic parasite, okay? Moving on to the next question, students. How do cytokine barriers provide immunity in humans, okay? Students, this also I have seen a lot of questions. I think many students might ignore this actually, thinking that why would they ask this? But this question also I have seen a lot of 1 mark, 2 mark questions. Cytokine barriers include interferon. So I'll give you a very simple example. Let's take the example in our class only. So let's say you people are all neighbors. Okay, you people are all neighboring cells. Okay, cell 1, cell 2, cell 3, cell 4 and cell 5. Okay, so A, B, C, D and E. These are the 5 cells that are there, okay? They are all neighbors, okay? They're all staying together happily, okay? They're staying together. Now, one villain comes into their life. The villain is none other than your virus. Virus comes and attacks A. Virus comes and attacks A. All of them were living happily, but virus came and attacked A. Now, students, we humans are not like this, okay? When some problem comes, we do not help another person. Instead, we put that person into a problem, right? But these cells are not like that. What this A is going to do, no? A is going to be like, it is going to release interferons. It is going to release what? Interferons, which are going to signal B, C, D and E cells, telling that A is being affected by a viral infection. You please protect yourself. Interferons are going to do the job of signaling these cells, and these cells are going to make antiviral, okay, antiviral antibodies and protect themselves. Understood? So, do humans do this? When someone is in problem, we'll be like, oh God, thank God he's in problem. Okay, we laugh and we mock. I'm not telling everyone are like that, but then there are some people like that, okay? But these people, the cells are not like that. This is called as cytokine barriers. Okay, I'm explaining the concept also, so we do not have to, make another class to learn the concept. Fine, cytokine barriers. So, interferons are going to signal. So, here you see, cytokine barriers include interferons which are secreted by virus infected cell. They provide protection to the non-infected cell from further viral infection. So, these full cells will be protected from viral infections. Clear, students? Easy peasy lemon squeezy. Okay, easy, no? Interferons, okay? This is your cytokine barrier. So, just write this. So, when I'm telling you, in both, just try to understand the concept. Okay, just understand the concept. Don't, be like, okay, I have to by heart it. You just know the words. You just know that cytokine barriers is interferons, which include interferons. Interferons are secreted by viral infected cells to protect the non-infected cells. Okay, that's all. All the keywords are there. The teacher will be looking for what? See, I have taught PU students. Okay, I have taught in Jyotinavas College. For those of you know, I have taught in Jyotinavas College, and I have spoken to many teachers who have, years of experience, like, 20, 23 years of experience, and they have guided me and mentored me during my, when I was teaching. So they have told me that, when they are correcting a paper, they only look for keywords. They just look for words like cytokine barriers, interferons, viral infected cells, non-infected cells. That's it. They do not want your, proper Shakespeare English there. They want keywords. Okay, so please keep in note. So both are very easy, students. All matter is if you score good in both, you can get good scores in KCET as well. Now look here, write the scientific names of pathogen used. So here you can see typhoid will be repeated. So like this only, malaria, like that, it continues. Okay, so let's go with the first one. All of you give me the answer for all of this. Yes, you can write the notes, Vinuta, or else I will be sharing the notes on this Telegram channel. Okay, I'll be sharing the notes on the Telegram channel. This is the Telegram channel, Biology by Gopika. Okay, Biology by Gopika, or else you can also, the main Telegram channel you have to join, that is V Karnataka, KCET and both. Same channel name, if you type, you will get. And it's in the description also. Okay, KCET boards. This is the Telegram channel. Fine? Yes. So students, moving forward, tell me the answer for this. So first we start with typhoid. Typhoid is caused by Salmonella typhi. So last class when I taught you human health and diseases, I made you people write one whole page of all the causative agents, right? Next one they have asked is malaria, that is Plasmodium vivax. Plasmodium vivax. Next what they have asked, amoebiasis. I'm not going to go into the typhoid, I'm writing this, okay? Amoebiasis, can anyone answer? Amoebiasis, give it a try. Pneumonia, give it a try. All of this I've taught you people, that's why you can try. Very good. Kush, Vinuta, Salmonella typhi, very perfectly right. Yes, try for amoebiasis. Very good, very good. Entamoeba histolytica. Entamoeba histolytica. Perfect. Okay. Next we have pneumonia. You can remember two of them. Streptococcus pneumoniae. Streptococcus pneumoniae and Haemophilus influenzae. Okay. Haemophilus influenzae. Got it? Next one is what, students? Common cold. Very good. Perfect. Common cold, ma'am. Rhinovirus. My friend is rhinovirus, right? Rhinovirus. Next, common cold and ascariasis. Ascariasis, very similar to the terminology itself. Ascaris. Okay, Ascaris lumbricoid. Ascaris, you would have studied this in your animal kingdom chapter. Okay, Ascaris. Then write the scientific name. You have filariasis or elephantiasis, which is caused by Wuchereria bancrofti. Okay, bancrofti. All of it done. Okay, perfect. Good job, Yasmin. Good job, Vinuta, Kush, Mohit. Very good. Okay, so students, Salmonella typhi, Plasmodium vivax, Entamoeba histolytica, Streptococcus pneumoniae, Haemophilus influenzae, Rhinovirus ascaris lumbricoid, and Wuchereria bancrofti. So these are the causative agents of all these diseases. And let me tell you, there is no assurity that question will come every time on typhoid. Anything they can give you. It is up to the board that they can give you any pathogen and you are expected to write it. Okay. Yes, ascaris lumbricoids. Very good. So that is done. Next question. See here, this questions also come. How does transmission of each of the following diseases takes place? Okay, so they have given here 1, 2, 3, 4 diseases. First is amoebiasis. So you know, most of the diseases are fecal oral route. That means from the fecal matter it comes into our oral route, right? So that is the cause, most of it. Okay, so here amoebiasis, students, disposable of the fecal matter causing contaminated. So here also it is contamination of drinking water and food, and this can be the carrier. Here could be your housefly. Okay, housefly can be a carrier. Remember, carrier and vector is different. This also I have explained to all of you in the last live that I took. Okay, housefly, that is a carrier. That means it goes and sits on the excreta and brings that to the home, and it lays on it, keeps its legs on the food item. Okay, yes. Next is, so houseflies and air currents transport the cyst to the pet animals as well. Malaria, female Anopheles injects sporozoids along with saliva. So here malaria will be caused mainly by your mosquito. Okay, ascariasis, students, again food particles, dirty hand, contaminated water, all of this only is the reason, by fecal itself. Pneumonia, droplets and aerosols of an infected person. So if you see, only these two are not in the fecal oro matter. Okay, fecal oro route. Malaria and pneumonia are mainly, like, it will be transported from a healthy infected person to a healthy person by a vector, and pneumonia, it is by droplets or aerosols. Like I sneeze, okay, and someone else also has pneumonia, they sneeze, that droplets or aerosols come in and we get the disease. Okay, or by exchanging something, now a pen or some handkerchief where the aerosols are there. Okay, during that cases spreading is going to happen. Now this question is being repeated forever. Like, I think this is, like, you know, board's favorite question. Draw a neat label diagram of an antibody. So this is the diagram that I drew in last class for all of you. So I've just taken a snip of that and added for all of you. So students, please request all of you to learn antibody diagram and go. I have seen so many questions on it, like in previous year, almost all the papers, even in mock papers, even in, you know, sample papers, pre-board papers, everywhere this question is one of the favorite questions. Okay, antibody diagram, because it's easy also. See, you draw 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 lines you draw, broken lines. Okay, so here one line, here one line, here one line, here again this same thing goes here. Here, students, very importantly remember that you have, what are these antibodies? To make the antigen, to bind with the antigen, right? So the region in the antibody where the antigen will come and bind is called the paratope. Okay, paratope. And then here antigen binding site is given in your NCERT. You can stick to this. You can write antigen binding site. Even if you forget paratope, it's okay. Write antigen binding site. Okay. And the most important thing is these are the light chains and these are the heavy chains. Okay. There are light chains and heavy chains. So antibody is made up of wait, here somewhere I've written. Yeah. Antibody is made up of 2 heavy chains and 2 light chains. Okay. 2 heavy chains and 2 light chains. This is how they are made up of. 2 light chains. And students, remember that they have disulfide bonds. Okay, disulfide bonds. These have come for 5 marks also. These have come for 3 marks also, Vinuta. Okay, antibody question has come in for 5 marks, 3 marks also. Okay, so disulfide bonds are there. How many disulfide bonds are there? 16 disulfide bonds are there. Students, even if you are not able to remember the number, no problem. Please write that antibody, draw the antibody diagram. Okay, carefully draw the antibody diagram. 2 lines here, 2 lines here, 2 lines connected to it. Always notice that these 2 lines are connected by a bond to these larger lines. Okay, and then these 2 are coming here. This is an antigen binding site which is called the paratope. Okay, and epitope is the region of the antigen that is going to come and bind with the antibody. Okay, so here you have the disulfide bonds, 16 of the disulfide bonds. Okay, 16 of the disulfide bond, 2 identical antigen binding site. Clear? Yes? Hi Rathesh. Okay, done. That is a very important question. Please try to remember as much as possible of your antibody when you're drawing. Just draw this, students. Okay, draw these 2 lines, connect it. Okay, draw like this, draw like this. Again break it, then draw here like this, here like this. Your antibody will be done. Even if you don't draw this S also, it's fine. Just connect it like this. Okay, connect it like this. That's all your antibody diagram is. Okay, and you write 2 heavy chain, 2 light chain. Right, it is bounded by disulfide bond. Teacher will blindly give you mark. Okay, trust my words. Okay, now define immunity, explain active and passive immunity. So students, immunity means what? The ability of the body to fight a pathogen, right? That is immunity, ability of the body to fight a pathogen. That is exactly immunity. So immunity means that. Okay, active and passive immunity means, remember, active immunity. Okay, active immunity. Here, remember, active immunity means we are talking about our body making the own antibodies. Okay, body will make the own antibodies. But when I talk about passive immunity, okay, when I talk about passive immunity, different story it is. Passive immunity could be ready-made antibodies. Okay, ready-made antibodies. Yes, ready-made antibodies. So, active immunity means own, our body itself, we are giving body time to make antibodies. Ma'am, how does this happen? Students, this happens in 2 scenarios, like vaccination. Okay, like vaccination, that means we have given weakened antigens, okay, or inactive antigens we have given, injected into our body, and we are telling the body that you please make antibodies, I will give you the time to make antibodies. This is one way of doing it, okay, where we are giving vaccination, or we give like we get an infection and our body makes antibodies against it, like, you know, acquired like any diseases that we have got, any preclinical situations that we have got, body recognizes it and starts making antibodies against it. These are the 2 active immunities. Passive immunity, when they say, could be one way like colostrum, that is the mother is giving to the child. Okay, colostrum. Students, colostrum already has IgA. Okay, it is rich in antibody IgA. So colostrum, but it is passive natural immunity. Okay, under passive and active there is natural and artificial. I hope you people know this. So here it is, to the mother we are not injecting anything, telling that please make antibodies for me. No, the mother is naturally making antibodies to give it to the child, right? To give it to the child. This is called as passive natural immunity. Then we have passive artificial, students, where like anti-venom, that is we inject venom into the horse or into slaughter any animals, and we get ready-made antibodies from it, and we will be injecting it to a person who has got snake bitten. That is anti-venom. Clear to all of you? These are active and passive immunity. Okay, active, passive, and active artificial, active natural, passive artificial, and passive, yes, antitoxins. Very good. Yes, this anti-venom we can, that is antitoxins. That means for any disease for that matter, if we have used another animal or anything and we have told them please make ready-made antibodies, then we take that antibodies directly. That is called as your passive. Okay, clear or not? Now, right, explain the life cycle of HIV. Do I even have to tell you people how important this two questions are? Explain the life cycle of HIV in the body and write a schematic representation of the life cycle. Students, you might think that how can this teacher ask me this question for exam? How can this teacher expect me to draw this diagram? But the teacher is not thinking that. The teacher is thinking that which question can I give for like 5 marks at one shot. So they are thinking that. Okay. So students, here is your HIV life cycle of HIV. But before you go into the diagram, I would request all of you to write this basic things. Okay, write that HIV's full form you write. Okay, full form. Then you please write that HIV affects cells of a, one second, HIV attacks cells who have CD4 receptor in them, right? CD4 receptor in them. CD4 receptor in them. And which are the cells who have CD4 receptor? I have only taught you this. Dendritic cells. Okay, if you haven't seen AIDS and HIV class yet, go and watch it after this class. It's only a 1 hour class. Dendritic cell, macrophages, and macrophages are the factory of HIV. Macrophages as well as your T cells. Okay, as well as your T cells. These are the 3 people who have CD4 receptor in them, and these HIV will be happy to go and attack these cells because they have CD4 receptor. HIV's glycoprotein 120, remember GP 120 will go and attach to this receptor and it will enter inside. So students, when you learn about virus, what is that one thing you learn about virus? That it will release its genetic material inside the host cell. Okay, here also same thing is going to happen. Look here. This is the so let's say it has bound to the CD4 receptor. It will release the viral RNA into the cell, into our host body. Okay. Oh, your teacher did not explain the concept. Okay. Okay, so now I'm explaining it. Okay, if you have any doubts, you can ask me. So remember students, human immunodeficiency virus. Now it will only affect the cells who have CD4 receptor. CD4 means cluster of differentiation. Okay, no need that much in depth. CD4 receptor. This glycoprotein, okay, glycoprotein, last time I had drawn the diagram. If all of you remember, students, this is the HIV. I cannot draw the whole diagram at this shorter period of time. Okay, this is your HIV. On this HIV, remember that this, like this glycoprotein is there. Actually, college, no one explains this much because it's out of syllabus. Like in the sense, to make a student understand, we need to explain this so the student will be able to relate it. Most of the students, they by heart and go. Right. So students, remember this is GP 120 and this is GP 41. These are glycoproteins. Okay. So this GP 120 will go and bind to the CD4 receptor and it will enter inside the cell. Now when it enters inside the cell, remember that this viral RNA is introduced into the cell. Very, very important thing for all of you to remember is that this HIV fellow, no, he is not very simple fellow. He has reverse transcriptase enzyme. What does he have? Reverse transcriptase. And students, this reverse transcriptase is going to make sure to convert viral RNA to DNA. Ma'am, why is this happening? Because students, we have DNA, right? We have DNA. Since we have DNA, this host, that is the virus, has RNA. This reverse transcriptase will convert RNA to DNA. Think it this way. DNA to RNA is called transcription. Reverse transcription means RNA to DNA, right? RNA to DNA. So viral RNA is introduced into the cell. Viral DNA incorporates into the host genome. Here you see, viral DNA is produced by reverse transcriptase. Viral DNA will now be introduced into our cell, okay, host cell. So host genome, new viral RNA. So inside the cell, the body will tell you know what? Central dogma, all of you remember, no? DNA to mRNA, mRNA to protein. So inside it will go to the host and it will be like, you make my proteins, you make my RNA. So viral RNA will come out, okay? So new viral RNA is produced by the infected cells, and that will lead to the production of new viruses, and new viruses will go and continue the multiplication. So remember that whenever HIV is going to affect, okay, whenever HIV is going to affect macrophages, it is not going to kill the macrophages. Instead, it is going to use macrophages as a factory. It is going to use macrophages as a factory. Very important for you to remember. And it is going to kill T cells. It is going to harm the T cells. And students, because they keep killing the T cells, a person who have AIDS will have less immunity. Clear? The concept is clear? Lot of questions have come from this topic, so it is important for you to understand the concept and remember the keywords, okay? KCET PYQs we will be doing in the month of Jan and Feb. Vinuta, Jan, Feb, March continuously, okay? Yes, students, tell me if the whole concept is clear. If you understand the concept only, you will be able to write a 5-mark question, a 3-mark question, because diagram you will be able to draw, because you can just draw one circle, draw an RNA, put the RNA inside, convert it to DNA, send it inside, produce the RNA, again produce new viruses. It's most easy. But writing the concept is important, that HIV is full form. Write that HIV affects cells with CD4 receptors like your dendritic cell, T cells and macrophages, and it uses macrophages as a factory to produce new viruses, but it kills T cells, indeed reducing the immunity, and an AIDS person will die out of, not because of AIDS, because of opportunistic diseases, okay? Dendritic cells are your neurons, we have studied, no? Dendrites, axon, all that, those are your dendritic cells, okay? Yes. Now students, macrophages, okay, you remember, very important. HIV has enzymes called as reverse transcriptase, which converts RNA to DNA so that it can enter into the host genome, okay? Yes, got it? Fine, that is the question. So explain the life cycle, you can explain it, write down the life cycle, and when they ask you to draw the life cycle, you draw the life cycle. Students, for a 5-mark question, don't draw and write the life cycle. Neither go for drawing or go for writing, okay? Don't draw and write, it will become too much work for you people. Yes, describe the life cycle of Plasmodium, that is your Plasmodium vivax. Please write the life cycle, okay? Life cycle, that is your asexual phase and sexual phase. The malarial parasite requires 2 hosts, that is human and mosquito, to complete its life cycle, where the female mosquito is the vector, okay? Female mosquito is the vector. Now you go back to, where is the, one second. Yes. Students, this question also, you know, no, very important, malaria question, so it will come. If they don't ask only, I'll be suspicious why they did not ask. So look here, this is your life cycle given in your NCERT. All of you open your NCERT in case it's not clear to all of you. So students, very simple, okay, you start with sporozoite. So an infected mosquito which has Plasmodium vivax will have the spore stage of it. It will come and bite a healthy person. in the healthy person, it will automatically go to the liver cells. The sporozoites will go to the liver cells. In the liver cells, it will asexually multiply and increase in number, and it will burst open the liver cells. It will burst open the liver cells, and obviously if something burst open, it is going to the blood only, no? It will burst open the liver cells and it will enter the RBC. In the RBC also, it will asexually multiply a lot. It will burst open the RBC. RBC will release hemozoin. Okay, it is given in your NCERT if you go and check. Hemozoin, which causes fever and chills in a person who is affected with malaria. This hemozoin also helps in favoring the formation of this gamete. Okay, so fertilization will not happen in our body. Gametophytes, that is female and male gametophyte formation, will happen in our body. That will be taken up by the next mosquito or the same mosquito as a blood meal. Okay, so when they take the blood from the infected person, they will get the gametophytes. Okay, they'll get the gametes. In the gut of the mosquito, fertilization will happen, and that will lead to the formation of sporozoites. Okay, now that I've told the whole story, let's just go through it. So look here, students, when the mosquito bites a person, the parasites or the sporozoites reach the liver, okay, liver through the blood. Now, in the parasite will usually reproduce asexually in the liver cells, and the cells bursting open the liver cells, and it will be released into the RBCs. In the RBCs also, it will burst open the RBCs and it will release hemozoin. Now, remember here, sexual stages, that is your gametocytic stages, develop in the RBC. So, sexual stages, that is your gametophytes, okay, will be formed in your RBC, but fertilization will not happen in our body. Only the asexual phase of Plasmodium vivax will happen in a host body. Sexual phase will happen in the mosquito's body. Okay. So, students, here female and male will be formed, that the female mosquito takes up the gametophyte as a blood meal, and fertilization will happen in the mosquito's gut. Okay, and then students, the sporozoites will be stored in the salivary gland. Mosquito is very smart, no? He's like, oh, if I go to bite, if it has to come from my stomach, it will become a long process, so I'll keep it in the salivary gland only, so I can go and poke every healthy person possible. Okay, do you want me to write a flow chart for this? Quickly tell me in the chat box if you want me to write a flow chart. I will write it in 2 minutes. %, okay? Yes. So, look here. Sporozoites. Okay, sporozoites will enter into a man. Okay, will enter into the liver cells. In the liver cells, it will burst open the liver cells. Okay, it will burst open liver cells and it will get into the RBC. In the RBC, it will burst open the RBC, burst open RBC by asexual. Okay, remember this, asexual stage is happening in man. Okay, asexual reproduction leading to the release, okay, leading to the release of hemozoin. Students, no grammar in this, only flowchart is there. Okay, leading to the release of hemozoin, and this hemozoin causes the fever and chills in a person who has malaria. Okay, fever and chills, and they will also help in the sexual stage, that is in the formation of the, okay, formation of the gametes. Gametes will be formed in the RBC. Clear? This is clear? Fine? Yes. Now, the gametophytes or the gametes, both the male and female, will be taken up as a blood meal. Okay, it will be taken up as a blood meal by mosquito. Which is the mosquito? Female Anopheles mosquito. Okay, by the mosquito, and fertilization will happen. Fertilization will happen in the gut of the mosquito. This is important, students, because your MCQ question can come asking that where is sporozoite stored and where is sporozoite fertilized. This question can come. Okay, gut of the mosquito, And then this will, obviously students, gametes fertilization will happen, so it will form the sporozoites. Okay. Sporozoites will be formed. Sporozoites will be formed and stored in the salivary gland. Stored in the salivary gland. Clear? Yes or no? Please copy this down. Okay, please copy this down so all of you can study it later. Please copy this down and give me a thumbs up once you're done. I'll take, wait ma'am, I'll take screenshot, okay? Done? Okay, I'll come into the, okay ma'am, continue. Okay. Ma'am, what if, students, see, if first of all, if Plasmodium has entered our body, it is going to cause a big damage to our body, okay? It is going to block our vascular systems, blood flow. So, if, in case, okay, in case, ಚೆನ್ನಾಗಿದ್ದೀನಿ, so in case it is there in our body, it is going to cause damage itself, okay? Because we are already infected with it. Plasmodium vivax has already entered our body. If gametes are there, then next mosquito comes, it will take in the blood meal or not, we are still going to get affected, okay Kush? Yes. So this is the whole flow chart of it. Now the next few, I saw a lot of questions like this with different diseases, okay, different diseases. See, a health camp was organized, can you read it? A health camp was organized in a village to conduct routine checkups for villagers. 5 patients with the following symptoms were recorded and preventive medicines were prescribed. Identify the diseases which are suffering based on the symptoms. High fever, 39 to 40 degrees Celsius, blindly you can write typhoid. Because that is the number specified, no, 39 degree to 40 degrees Celsius, blindly you write it, okay? Chills and fever recurring every 3 to 4 days. Students, this is a symptom of malaria. Why this is a symptom of malaria? Because your hemozoin should be released. If hemozoin is released, only the person will get fever and chills. So until that life cycle happens and hemozoin is not released, fever and chills symptoms will not come. So every alternative to 3 to 4 days, this person will get fever and chills. Clear? So that is why here they are telling chills and fever recurring every 3 to 4 days. Bouts of fever and diarrhea is the nasal congestion and discharge, common cold. Lips and nails turn grayish to blackish is extreme fatal case of pneumonia. Okay, so pneumonia, I feel this is easy to identify, right? This is easy to identify. This is easy to identify. This is easy to identify. This you might get confused, okay? Because AIDS, bouts of fever and diarrhea, just remember that continuous, okay? Even in Midson, the same question was present. Exactly. Okay. Nasal congestion and discharge, student, this you can identify. This also you can identify. Only this you remember that fever will keep coming and the person will also have diarrhea, that is AIDS. Okay. AIDS. Clear? Next question. List any 2 methods of HIV transmission. So students, some more questions that keep coming is about drug abuse. Okay. Drug abuse. How we can do prevention, okay, prevention, transmission, all based on this only prevention comes, transmission. They are all, you know, very common, what is that? Like how we talk to each other and tell, no, don't do this, don't do that. It is more of that. That's why I have not picked up the questions. Like, you know, peer pressure, reasons for drug abuse, transmission of drug abuse, okay, how we can get rid of drug abuse, talking to a counselor, asking a mentor, having good friends, all that is general things that you can write and teachers will blindly give you marks because there's nothing to write from, like, you know, from all the brain cells that we have. So they will give you marks, okay, don't worry about that. Now, next question is, students, list any 2 methods of transmission, okay? Sexual contact with infected person, transmission of contaminated blood and blood products. Right now only we did one more question of this. So they can ask you two methods, three methods. First thing, contact with the infected person, sexual contact with an infected person. Second, blood transfusion. Third, from the mother to the child through placenta. And then you can have by intravenous drug abuse or by tattoo. Okay, or by tattoo, but tattoo is not mentioned that commonly, but intravenous drug abuse is mentioned. Sharing needles and syringes by intravenous from infected mother to her child through placenta. Individuals with multiple sex partners or sexual partners. Okay, during this stage also AIDS will come. Any doubts in this? Easy peasy, this is all. Now, what is contact inhibition? What is interferons? Mention the role of alpha interferons in the treatment of cancer. Very good questions. These questions are from that, that Karnataka PUC board gives the handbook, no? These questions are from there, okay? Yes. Students, what is contact inhibition? That is a property of the cell to stop cell division when it comes in contact with another cell. Cancer cells will lack this, okay? So it is the property of a normal cell by which their uncontrolled growth stops when they come in contact with another cell. Okay, when they come in contact with another cell, they will stop the cell division. That is understood, no? Like I told you the example of how Yasmin and Kush and Riyaz were sitting. They understood that they have only this much place, so they will not move or push each other. They will just sit in that place. That means when they came in contact with another person, they got to know, okay, I have only this much place, I need to stop. Okay, yes. Next is, what is interferons? I told you interferons are released by viral infected cells to protect non-infected cells. This is like helping each other, okay? Helping each other. Yes, this is enough for both, dear. Like, I'm not telling this one session is enough. Like this, similar questions are enough, okay? Similar questions are enough. You have to do the studying part, okay? Now you know the questions. Now studying is more important, okay? Yes. So antiviral proteins produced from virus infected cells are called as interferons. These are released so that they can protect the non-infected cells. Protect the non-infected cells. Students, I would say for both practice questions, okay, if you know the concepts, if you know all of the concepts, like at least like 80% of the concept, let me tell you, please stick on to your NCERT, especially for maths. Okay, maths, because I have experience. I was studied in PUC board here in Karnataka only. I studied in Christ. So I know that the last few months, that is the last 2-3 months, I only practice questions from handbooks and from previous year question papers, and 80 to 89% of the question was repeated, because every time they are not going to modify the question, only numbers will be changed, pattern will be changed. That way PUC board is the best. Okay, questions will be repeated, but let me tell you, the catch in PUC board is that they will not correct your paper like ICSE board. So I was in ICSE and then I went to PUC. Okay, so what happens is, students, there in PU board, what happens is they will not check, like they will only look to the keywords, because they will have an answer key and they will have your answer sheet. So when they look in the answer key, they will notice that, okay, this word is missing, they will cut the marks. That is the only problem. But ICSE, it is not like that. They will read through every line what you have written, because it's more conceptual and other thing, application level. But PU board only stick to the answer key and the keywords. So please, requesting all of you, even in your midterms, even in your pre-boards, make it a practice to underline the keywords. Take 5 minutes. 5 minutes time in the last, underline the keywords, especially in biology. Okay? Yes, Vinuta, I do know Kannada, okay, but I am not talking fully because many students do not understand, okay. Yes. So here, students, mention the role of alpha interferons in the treatment of cancer. It's a biological, treatment that is done where we use alpha interferons, again, for the body to recognize that, okay, virus has come in and I have to, protect myself. Activates immune system and helps in destroying the tumor, okay. So keep this tip with you. Please underline the keywords and thank me later for it. Let me tell you, I have experience with PU board. I have also been to the PU board, okay, for one of my friends, this one, and I saw the situation there, and I saw how the paper correction, how they are expecting out of a student. Okay, we cannot blame them also, because they have so many papers to correct every single day, and different teachers from different colleges, different perspectives come together and correct the paper. So it is your job to make sure that you're making the teachers' work easy by underlining the keywords and writing only to the point. Don't think that, oh, PU board is there, so I will write, Pushpa 2's full song, okay, or I will write, KGF ka story. No one is going to read. No one is going to read, okay? Yeah, please make a note of that. Fine. Now, important questions, students. So, we are coming to an end of this class as well. Important questions, okay? Yes, ma'am, we will underline the keywords. Good job. So, mention the 4 barriers of innate immunity. So, students, innate immunity, just remember that you have your physical barriers like your skin, okay, which are going to protect you from most of the pathogens. Then you have physiological barriers. Physiological barrier, it's given clearly in your NCERT, okay. Then you have cellular barriers like your natural killer cells and all of that, okay. And then you have cytokine barriers, that is your alpha interferon, okay. Skin, mucus lining, okay, mucus lining in your digestive system, respiratory system, all of those people are protection, okay. Then you have your cells like your natural killer cells who are going to do the job of your cellular barrier method, cellular barrier protection. And then you have cytokine where your alpha interferons are going to come in place, okay. Then explain the structure of antibody. I told you this is a favorite question of PU board, so please remember this. Distinguish between benign and malignant, benign and malignant tumor, students. Benign tumor is confined to one location, okay. Last time also I told you benign is confined to one location. It will not move around everywhere. It will stay where it is, and it is not harmful every time like the malignant tumor, and it can be easily removed, okay, because it is confined to one place, okay. And it is not necessary that it becomes cancerous as well. Malignant tumor on the other hand is not like that. Malignant tumor will spread everywhere, uncontrolled division. It will cause metastasis, that means from one place to another place it will run and go, and it will make tumor there also, okay. That is the difference. Now, what are carcinogens? Mention any 2 groups of carcinogens with one example is. Carcinogens are, active ingredients which can cause, like, we can say carcinogens are substances or materials that can cause cancer, okay, that can become potential cancer causing people, okay. Okay, yes. So the carcinogens are the ones that causes cancer, and carcinogens can be your physical carcinogen, chemical carcinogen. That means, students, UV rays, X-rays, ionizing is there, non-ionizing radiations are there, okay. UV rays, X-rays, gamma rays, very physical agents, physical carcinogens. Then you have chemical carcinogens like your tobacco. Okay, this 2 things you can remember easily, don't even have to write separately. You just write physical carcinogen and chemical carcinogen, right under that only UV rays, gamma rays, X-rays, they'll give you marks. If you have confusion which is ionizing, which is non-ionizing, just write all of these 3 points under, okay. I'm giving you a lot of tips here because I want all of you to excel in your boards. And students, next is biological carcinogens also you can write, that is your proto-oncogenes, okay. In our body there are cells called as proto-oncogenes. Students, this proto-oncogenes, when they are exposed to your X-rays, gamma rays or any carcinogen, they will automatically become cancerous, okay. They'll become cancerous. This is your biological carcinogens. Proto-oncogenes are normal cells, like it is present between our normal cells, but when they are exposed to extreme UV rays, gamma rays or any other physical or tobacco smoke, they will become cancerous. They will become oncogenes, okay. Yes, carcinogens are substance or any potential ingredients that can cause cancer to you, okay, that can become cancerous, okay. That is, they have the ability and capability to cause cancer. Carcinogens are of 3 types, okay, physical, chemical and biological, okay. Physical are, students, your X-rays, gamma rays, if you are exposed to this, UV rays, these 3 people will come under this, okay. Chemical, you have your smoke, that is your tobacco, and biological is your proto-oncogenes. These are normal cells, but when exposed, okay, to any of the physical or chemical agents, these people will become problematic, okay. Now, briefly, is clear? Fine, Yasmin? If it is clear, I'll proceed, or else I'll explain again. All of you drink some water, hydrate yourself. Okay. Briefly explain 3 prevention and control measures. Yes, this is the question I was telling all of you. Students, this question also they have asked, and most of the toppers answers in drugs. Please try to learn opium, okay, cocaine, coke, crack, all that. Just understand which is how they are, you know, from which plant are they, okay, is it from papaver, is it from, you know, is it shoot, stem? Just know that much. Just know that much. It's more than enough. Okay, more than enough. Okay. Yes. What did I send in this? Yes. Clear? Fine. The notes will be on the Telegram channel. Don't worry at all. Okay. So, students, this question also I have seen, okay, some places, that is name the disease. Okay, name the disease caused by following organisms. Like they'll give you rhinovirus, they'll expect you to write what is the disease, or they'll give you this missing, this missing, any one of it they can give you. Okay, prevention is basically talk to a counselor. Okay, like do not hang out, like do not go around with people whom you know as drug abusers or alcohol abuse, and then, you know, avoid peer pressure, avoid, you know, having going to conversations where you feel low and other things. So it's very general things. Okay, general things, like you can just imagine, no, why do people take drugs? So that they're not in the reality. They want to escape the reality and they want to feel that, okay, everything in my life is sorted. But then the next day, everything will be back to normal. They wake up their eyes, they'll be like, oh my God, I still have 0 bank balance. I still have to go for an exam. So everything will be back to normal. So people do drugs just to avoid, or, you know, get rid of that moments, that pressure at that moment. But nothing is going to change in life, everything. So that's why people have a tendency to go back over and over again, because they do not like the reality. So this happens because of peer pressure, that you want to excel exactly like your friend there, or you want to do, you know, you want to be cool, especially in teenage, you know. So 15 to 25 years is the age which maximum people do drug abuse and alcohol abuse. Okay, students? Students, this kind of questions, as I told you, they can give it as match the following. Okay, it's a very good question. Can come as match the following. Okay, match, missing, or 1 mark. Okay, or 1 mark. It can come as any of this. So please be prepared for this. This is your whole box. I've put this box so that when you're studying, no, you can just look into this box and study. Even last time when I shared the notes also, I had given you that snip of it. Okay, done and are done? Yes. So that's it for today's class. We have done the most repeated questions. Okay. Oh, is it? Oh, I have to check that. Okay, I will check that also. So students, we have done our, when is the dates, Kush? Is it 18th and 19th? When is the dates? Okay, so that's it for today's class. I hope all of you have enjoyed the most repeated questions from human health and diseases. Please be regular to class and try to, you know, be till the end. I know, students, the journey is not easy, the journey is long enough, but to be in a good lifestyle, to earn well, and to make your parents proud, to make yourself proud, to even buy something for yourself, okay, to buy something for yourself, we need, okay, we need it. Yes, 18th and 19th only. Okay. So you need to get into a good place, you need to live life, and you need to earn money also as well. Money is very, very powerful. For that, you need to land in a good job. People might tell, no, you have to not do it for money, but I wouldn't say that, because we all are working and we are all working for that. Okay, thank you so much for joining. Please take 1 minute of your time and drop a comment below to let me know how the class was, because it will boost my energy, and that is the only thing I'm asking from all of you, nothing else. Like the video and comment down how you're feeling about the classes and how you enjoy my classes, so that I can bond with you and I will also know your names, and we will have a very good connection and very rapport in the future as well. Okay, so thank you so much for joining. See you all in next class. See you all. Take care. Bye bye.